Hello students. Today the topic which we are going to start is another blood cell, that is a platelets. I think last year or in couple of years dengue fever was very popular, and every time whenever we are discussing with the people about dengue fever, and everyone is suffering from one issue, that was the platelets count. So today we are going to understand what is platelets and what is the importance of platelets. In some of the book it's mentioned that platelets is known as thrombocyte, but basically there is a difference in platelets and thrombocyte. Platelets is a term which is used only in case of human being or mammals, while thrombocyte is a term used in case of all vertebrates other than mammal. So platelets are present in mammal and thrombocytes are present in vertebrates other than mammal. Now first understand what is the importance of platelets in our body. Platelets is a very important part of our body because it helps in blood clotting. Whenever we have any kind of bleeding, that time the blood is controlled with the help of platelets. Now the question is, if any part injured and from that part if bleeding is there, then why it is important to control that bleeding? Just because that we want to prevent the loss of blood? No. There are two aspects of control of bleeding or two aspects of blood clotting. One is definitely we would like to control bleeding, and the second most important is we like to control the entry of microorganisms inside the body. Because suppose if this part is injured from here, then bleeding can occur from here, and moreover now this part is open, tissues are exposed, microorganisms can enter into the body easily and they can affect our body. So blood clotting is very important. There are few diseases in which people are not able to control blood clotting, like hemophilia. Hemophilia is a disorder in which people are not able to control their bleeding, even it is a very minor bleeding is there. Anyway, what are the characteristic features of platelets? They are enucleated in mammals, and in case of other vertebrates other than mammal, it is nucleated. So if I am talking about human being, in case of human being, it is enucleated. Enucleated means nucleus is absent. It is oval in shape. Size is very small. It is 2 to 3 micron. And what is micron? 1 micron is equals to 10 raised to power minus 6 meter. Okay. And what is the count? Count is approximately 2.5 to 4, or in some of the books it is mentioned 3.5 into 10 raised to power 5 per millimeter cube approx. Whenever you are writing the number of any of the blood cell, please write down the term approx, because it is not a very particular number. What is the lifespan? The lifespan is again very short, that is around 10 to 12 days. Now, what is the difference between thrombocyte and platelets? Thrombocyte is nucleated. So, in case of human being, in case of mammals, there are two blood cells which are enucleated. One is RBC and one is platelets. Only WBC is the blood cell which is nucleated, and rest all of these are enucleated. Now, the important function of platelets is blood clotting. Yes, like WBC, they can also do phagocytosis. like they produce pseudopodia. Pseudopodia means, what is the meaning of pseudopodia? Pseudopodia means false feet. Pseudo means false and podia means feet. They are able to produce false feet, and because of this false feet, they are able to do phagocytosis. Clear? Now, what we have to understand, what is the process of blood clotting. First of all, what is the scientific term for blood clotting? It is a very important question. Blood clotting is, or blood clot, scientifically known as thrombus. Scientifically, it is known as thrombus. Now, what is the definition of thrombus? Thrombus is a network of fibrin. Fibrin का network होता है, network of insoluble proteins, that is fibrin. Fibrin is one of the protein which is insoluble, in which the formed elements, what are formed elements? WBC, RBC, formed platelets, formed elements are trapped. So, what is blood clot? Scientifically, blood clot is no word. It is thrombus, and it is a network of insoluble protein fibrin in which the formed elements are trapped. यानी कि insoluble fibrin का एक network है जिसके अंदर formed elements like WBC, RBC, platelets are trapped. That is called blood clot. एक lock जैसे बनता है. You might notice, suppose this part is injured. This is your hand. Okay. And the muscle of this part is ruptured. Now, from this reason, platelets will release, and these platelets will release a protein that is called thromboplastin. अभी हम समझेंगे कैसे. Okay. Now, this clotting process occurs in 3-step process. Okay. Now, how this process occurs? Now, look here. First, when this part injured, from this part platelets release thromboplastin. Platelets release thromboplastin. So thromboplastin is the main constituent which start blood clotting. Now, in presence of this thromboplastin, there is another protein that is called prothrombin. Prothrombin converts into thrombin. It means thrombin. Okay. So, platelets release thromboplastin. In presence of this thromboplastin, prothrombin converts into thrombin and calcium ions are required. Okay. Why this calcium ions is required? Calcium ions act as a promoter. Okay. In presence of this thrombin, this prothrombin, first of all understand here, prothrombin, it is an inactive form and it converts into thrombin, that is an active form of protein in presence of thromboplastin and calcium ion. Now, in presence of thromboplastin, the third step is fibrinogen. Fibrinogen, it is a soluble protein. In presence of this thromboplastin, it converts into fibrin, and fibrin is insoluble protein. So these are the 3-step process of blood clotting. Okay, platelets release thromboplastin, which acts as an enzyme, and in presence of thromboplastin, prothrombin, which is an inactive form of a protein, converts into thrombin in presence of calcium ion, and in presence of thrombin, as well calcium ion. Fibrinogen, which is soluble protein, converts into fibrin. Now, how the blood clot will form? Look here. Suppose this is the part which is injured. Okay. Now, from this area, platelets will release. They will release thromboplastin, convert prothrombin into thrombin, convert fibrinogen into fibrin. Now, these fibrins make a mesh, make a network like structure over this wound. इस घाव के ऊपर ये स्ट्रक्चर बनाएंगे that is known as fibrin का. Okay, and all the formed elements like WBC, RBC, platelets, whatever is ruptured, they will gather here and they will lock this entire tissue. इसको पूरे को क्या कर देंगे? Lock कर देंगे and prevent the entry. It will form a like of a plug. अभी तो ये बहुत खूबसूरत लग रहा है, but actually it is not like that. It forms a like of dark black color coating from where the blood cannot come out. It makes the clot. It make lock the bleeding. It make a plug-like structure to prevent the blood loss or to prevent the entry of microorganisms. Now, here lot many important questions are there. First question, one more most important thing is all these processes require vitamin K. There is one vitamin which is required for blood clotting, that is a vitamin K. First question is write down all the steps for blood clotting. Simple question for 2 marks. Second question is name the, now listen very carefully, and many of the students do this mistake. Name the formed elements, name the elements of the blood which help in blood clotting. That is a thromboplastin which starts the blood clotting. Without thromboplastin it is not possible. Second, name the mineral which is required for blood clotting. That mineral is calcium ion. Third question is name the vitamin which is required for blood clotting. That is a vitamin K. Vitamin K try to stop the clotting at every step. First of all, it try to stop the process release of thromboplastin. But in any case, if thromboplastin is already released, so it will try to stop the formation of thrombin. In any case, if thrombin is also formed, okay, then they are definitely stop the process of fibrin. So they try their best to stop the bleeding at the time of injury. So I have discussed almost all important questions from this topic. याद रखिए, whenever you are writing the steps for blood clotting, you have to mention the presence of calcium ion. It is mandatory. Okay, now the question is why blood does not clot inside our body. Keep in your mind that blood cannot clot inside the blood vessel. Never, never, never. Inside the blood vessel, blood cannot clot. Why blood does not clot inside our body? Because of the presence of heparin. You might remember in 9th standard, I think, we have done the function of liver, and in the function of liver we have discussed that liver produce heparin, and heparin is anticoagulant. Now what is anticoagulant? Heparin, it is also a kind of a protein which is present in our blood, act as anticoagulant. Anticoagulant. Anti means opposite, against. Coagulant means clotting. It prevents the clotting of blood inside your body because of the presence of this heparin. Now you might be thinking that how we can store blood in our blood banks. We can store blood in a blood bank for few hours or even for the few days. This is because of the presence of sodium citrate and sodium oxalate. Sodium citrate and sodium oxalate. We add these two substances. we add these two substances in the bottles where we store blood in blood bank to stop the blood clotting. What they do, how they stop the blood clotting? They lock calcium ions. They lock calcium ions. And already we have discussed that calcium ion is required for the blood clotting at every step. Whether we have to convert prothrombin into thrombin, or we have to convert fibrinogen into fibrin. Okay. Now, last and important topic for this topic is, you need to understand two terms. One is fibrinated blood and one is serum. Defibrinated blood. Defibrinated blood. Okay. Now it is very clear from here. Defibrinated blood means D means no, निकल चुका है, and fibrinated means the blood which does not have fibrin, means blood without fibrin. Blood without fibrin is required for certain medical processes. How we prepare this blood without fibrin? Suppose this is a container where blood is stored. Put one stick and stir it. Okay. Then when you stir it, all the fibrin will collect on this string. This is fibrin. So whatever is left, this is a defibrinated blood because fibrin is already stick on this. जो भी stick हमने लिया उस पे जाकर चिपक जाएगा. Whatever is left is defibrinated blood. Now what is serum? Serum is blood without fibrin and blood corpuscle. A blood without blood corpuscle, that is no RBC, no WBC, no platelets and no fibrin. That is known as serum. Clear? Or we may write serum is plasma without fibrin. Now what is plasma without fibrin? How it is different or how it is common? What is plasma? Plasma is the blood without blood cell. So blood cell हमारा already यहाँ पर है, and this is plasma without fibrin. So fibrin is also not there. So serum is blood without fibrin or blood without plasma without fibrin. Okay. Now on all these bases we have four different kinds of blood group, and that we are going to discuss in next video, that how many kind of blood groups we have and which blood group can donate blood to another party or from whom you can take blood. Okay, till then keep revising, keep learning and keep asking, and take care.